Hello, everyone. Welcome to this second podcast for Chapter 12. In this podcast, we're going to be covering Objective 3 from your note-taking guide. In this, uh, in this objective, we're going to focus on the cell cycle and mitosis. More efficiently said, the mitotic cell cycle. In general, what we're going to ask is, how do we start off with a cell like this, say a diploid cell, which means it has 2 copies of every chromosome. In humans, that would mean we have 46 chromosomes. And how the cell goes through this mitotic cell cycle. To give us 2 identical cells. That's the key to mitosis, is that at the end of it, we end up with 2 cells that are genetically identical to the parental cell here. This is really important. For instance, if we had a kidney cell here, if we want the progeny cells to also be kidney cells, it's really important that they maintain an identity to this original cell that started the cell cycle. When this fails, and we'll talk about this in a later chapter, when this fails, we end up with various kinds of diseases, including cancer, but not exclusively cancer. All right, let's go ahead and move to the next slide and start breaking down mitosis in all of its glory. Okay, let's begin our discussion of the cell cycle by breaking down interphase. Let's begin by drawing a cell here. We'll draw it right here. And this cell is a new cell. It just came from the previous cell cycle. And what I want to do is draw some chromosomes in here so we can follow the chromosomes as we go throughout the cell cycle. I'm going to draw 4 chromosomes or 2 pairs. So this might be, say, chromosome 1 right here. It's a long chromosome, so we'll make this a longer chromosome. And then let's draw a chromosome, the second chromosome here. Okay, now that we have drawn the chromosomes here, let's review some terms. And let's just pretend we're dealing with these, what we see here, these 4. We know that we have 4 chromosomes. We could call these either 4 chromosomes, or we could call them 4 chromatids. Either would be fine. But for ease here, I'm just going to call these, we're going to say it's 4 chromosomes. And we would also say there are 4 chromatids. We would also call these 2 here homologous chromosomes. The 2 red ones are homologous chromosomes, and the 2 blue ones are also homologous chromosomes, meaning one of them initially came from one parent and the other came from the other parent. The cell then, as it begins the cell cycle, it will enter into a phase called G1. G1 stands for gap, G-A-P. What happens during the gap phase are a few things. We'll write 1 or 2 here. It grows, and it will also check for mutations. So I'll make a check mark here, check for mutations. This is really important because the last thing you want to do is copy a cell that has a whole bunch of mutations in it. Now I'm going to draw this next cell here a little bit bigger for a few reasons. One reason is it just grew a little because it went through G1. And the other reason is I need to draw more chromosomes in here. Now I keep referring to these as cells, but recognize if we're talking about a eukaryotic cell, which we are in these examples, like a human cell, this is all occurring in the nucleus. So this next phase we call S phase. During S phase, what happens is the DNA is duplicated through the process of replication. So what's happening here is replication. So let's go ahead and draw what our chromosomes are going to look like now. Our chromosome 1 here, the red one, remember each strand up here, I should say each chromosome here is going to duplicate. So this one here on the left is going to copy itself, and they're going to be connected at the centromere. And this strand here will also copy itself. The lengths are the same. Again, my artistic skills perhaps don't allow me to draw the same lengths each time, but the lengths don't change. Now let's look at the blue one. Each one of the chromosomes copy themselves. They're connected by a centromere here, just like so. So let's also now come back and ask how many chromosomes and how many chromatids we have. Remember, if they're connected, we call them a single chromosome. As long as they're connected, they're one chromosome. So if we count them here, we have 1, 2, 3, 4. We have 4 chromosomes. So let's write that down here. And how many chromatids do we have? This is a little trickier. The chromatids are the number of DNA molecules we have here. So we have 1, 2, 3, 4, 5, 6, 7, and 8. So we have 8 chromatids. Another review term here, just to remind you so we don't forget things we learned on the last podcast, is that these two together we call sister chromatids. These two together that are being held together by the centromere are sister chromatids. These are sister chromatids. And these last two blue ones are sister chromatids. We call them sisters to remind you because they are identical copies of the previous of the original strand here. Now the cell is going to enter into G2. And again, we have our same cell. In fact, the cell doesn't change any between S phase and G phase, except that it grows a little bit. G2 also stands for gap. I guess we can write that here, gap. Just like on the previous gap, what we're looking here for is to make sure that this cell is ready to enter into the next and last phase of the cell cycle. So it grows a little, it checks for mutations, make sure there's no mutations that occurred in the S phase, and then let's draw our chromosomes again here. So the two red ones stay the same. Nothing's happened here yet. The two blue ones are the same. And I'm not going to write it again. You can write it in your notes if you like. We still have 4 chromosomes here and 8 chromatids. Now what happens next here is that the cell, after G2, enters into M phase. And for now I'm just going to draw this box here, because a lot of things happen in mitosis in this M phase here. That's going to take an additional page or two to go through. Alright, so let's review real quick from this slide. What we have is the cell cycle divided into interphase and M phase. Interphase is composed of G1, which allows the cells to grow and it checks for mutations. S phase is when the chromosomes duplicate or undergo replication. And then it enters G2, where the cells grow a little more, and then it also checks for mutations. And then it enters into mitosis. And what's going to happen in mitosis, as you will see on the next couple slides, is that these chromosomes, these two red ones, will pull apart and move into opposite ends of the cell. At the end of mitosis, what you'll see are 2 cells being produced through a process called cytokinesis. I'm going to write that over here, because I think I have room over here. So that would go over this line here. Through the process of cytokinesis, we now have these 2 cells. They will consist of 2 of the red ones, 2 of the blue ones, and they're identical to each other. And they're identical to the one that started. And when I say identical, I mean genetically identical. I think the best way to learn this, like a lot of things in this class, is to sit down and draw this out. And draw it out many times, labeling it like I've labeled here. Think about how many chromosomes or chromatids are in each section. And then I think memorizing this will be a little easier. It is a lot of stuff, though, and I do recognize that. It's not simple. All right, now let's move into mitosis and talk about how we're going to go from this cell to these two over here. All right, let's talk about mitosis. Mitosis occurs after interphase, so after G1S and G2, after the DNA has been copied, and mitosis's job is to segregate the chromosomes. Let's begin by looking at what the cell looks like during interphase. So here's the cell and here's the nucleus. The nucleus, let's see, how can I best draw this? Let's draw the chromosomes like so. I'm trying to attempt to draw these in a very kind of stringy manner, because at this point the cells are not condensed, they're actively being copied or they're turning genes on, and because of that they're not visible under the microscope. It's not until the cells enter into the M phase or mitosis that the chromosomes begin to condense and they're now visible under the microscope. Okay, so I'm going to write the letter I here because this is interphase. This is what the nuclear material would look like during interphase. The first part of mitosis that we should talk about is something called prophase. Okay, so let's draw the nucleus here, and let's draw the chromosomes in the nucleus. And remember, it's gone through S phase, so it's been copied, so let's connect these two together. And for simplicity's sake, I'm only going to draw a single chromosome. So remember what this would have looked like in an earlier stage. If you could have pulled these apart, what they would have appeared like would have been, before it's gone through S phase, it's just something like this inside of the nucleus. Two single chromosomes like that. But they go through S phase, and they're copied, and now they look like this. In prophase now, the chromosomes have begun to, what we say, condense with each other, and now they are visible under the microscope. Another thing that's happening, and we can draw these on the other cell too, are these structures called centrosomes. I'm just going to draw them like this. As the cell begins to enter prophase, these centrosomes will now move over here, and they will begin to send out and form these spindles. And these spindles are microtubules that attach to the centromeres of the chromosome. Something else that's happening at this stage is that the nuclear envelope begins to break down. Okay, so it's starting to break down in prophase. And by the time we get to this next phase, I'm going to draw this a little bit bigger just so I have a little bit more room. The nuclear envelope has completely broken down. So I'm no longer going to draw the nuclear envelope anymore. Functionally, the nuclear envelope the nuclear envelope doesn't exist. But let's continue to draw our chromosomes because they certainly exist. And again, I'm only drawing one chromosome here just for simplicity's sake. They're still connected together by this centromere. Now the centrosomes have now moved over here, and their spindles are attaching just to one of the sister chromatids. We call this phase metaphase. And some of the hallmarks of this phase, let me explain to you, include the final breakdown of the nuclear envelope, but that's really something designated prophase. But the main part here is that the chromosomes have lined up along the center of the cell. The spindles have attached themselves from the centrosome to the chromosome at the centromere to just one of the sister chromatids. Essentially what they're doing is they're grabbing hold onto it, and we're going to see in the next phase what happens is they're going to begin to pull those sister chromatids apart. So let's talk about that next. So here's our cell again. It doesn't really get bigger, it just gets bigger because it makes it easier for me to draw, but in reality at this point here they're not getting bigger. And we've entered the phase now called anaphase. During anaphase, these two sister chromatids have separated from each other. So now we have something like this. And the reason they've separated and begun to be pulled to the other side of the cell is because the centrosomes have begun to pull their spindles towards this end. So it releases them from the their fellow sister chromatid, and begins to reel it in, pull it in to the one side of the cell. As it's doing this, the cell will begin to elongate a little bit. And that's as it prepares for the final phase here, called telophase. And I'm going to do my best to draw this here. Not too bad. During telophase, what happens is the chromosomes now are fully pulled to this side of the cell. And then what happens is that the nuclear envelope begins to form around here again. A very important part of the M phase, or the mitotic phase, is that the nuclear envelope forms around the just copied and segregated chromosomes. Also, something that happens is that these spindle fibers that were present in anaphase are now gone. They're no longer present. However, they do maintain the centrosomes. These structures here that will, when this cell divides on its own, will send out the spindle fibers again. So this is called telophase. Now the final stage, your book clumps in with telophase, so we'll do that too. But remember, some books consider this last phase to be a stage on its own. And this stage is cytokinesis. So mitosis separates the chromosomes, the nuclear material. Cytokinesis, what we're going to find is it separates the cytoplasm. It will break this down and form two new cells. So that's what we're going to draw here. Two newly formed cells. Each of these newly formed cells contains that same nuclear envelope with the chromosomes in it. I didn't necessarily mean to draw this super small, but they're the same size. With the chromosomes in it, the centrosomes will still be in here. Okay. At this point, the cell cycle has completed. And this cell will now enter the same cell cycle. It will enter G1, S, G2, and then go through meiosis again. And so will this cell. Okay, a couple of points I want to make here before we close this podcast is that what we've just described here is what we see for eukaryotes. The chromosomes condense, the nuclear envelope breaks down and it reforms. So these are clearly eukaryotic events. Now, bacteria undergo a very similar process. They also go through something very similar to G1, S, and G2, where they copy their chromosomes during S phase. But then what happens with bacteria is they just kind of pull apart and split in the middle and pull the chromosomes to opposite ends. But we don't have this elaborate setup in bacteria. And in bacteria, we call this binary fission. And the interesting thing about this is that bacteria, which are much older organisms, say, than most eukaryotes, well, all eukaryotes, what's interesting is that the same enzymes that are necessary for binary fission in bacteria are the same ones, or very similar ones, found in eukaryotes during the cell cycle and mitosis. One final thing I want to, well, I guess there's two more things I want to make sure I say. The next one is that what we've drawn here, even though it looks kind of complicated, you have to remember it's even more complicated, because if this were a human cell, I only drew one chromosome pair, or ultimately, two chromosomes. But if you have 46 chromosomes, you have to understand that this is really a traffic jam in the cell. All 46 chromosomes are being attached to spindles, are being lined up during metaphase, are being separated during anaphase, and then are being packaged into new nuclear envelopes in telophase. And this has to happen at a very coordinated and efficient manner in order to make sure that these new cells have a full complement of all the necessary chromosomes. The last thing I want to talk about is, as you're studying this and the previous slide where we walk through interphase, I encourage you to draw this out many times. This is the best way to learn it. And as you're drawing it out, ask yourself, what are the chromosomes doing? How many chromosomes are in each of these cells? In fact, maybe we should do that real quick, just so you know. Also ask yourself, What happens if prophase fails? What happens if metaphase fails? What happens if anaphase fails or telophase? What would the result be if any one of these events failed? So, as I just said, let's go ahead and count chromosomes. Okay, during interphase, if this was the number of chromosomes we were dealing with, one pair of chromosomes, one pair of chromosomes or two individual chromosomes, we would say during interphase, this organism would have two chromosomes. During prophase of mitosis, there would be two chromosomes. Over here, there's still two chromosomes. Now, we see something different now because now the chromosomes have been pulled apart and the centromeres have been relaxed. And so what we see here are four chromosomes. Then as they form around their nucleus, we see two chromosomes in this cell and two chromosomes in this cell. Now on your own, ask yourself, how many sister chromatids are found in each cell? That number is going to be a little different in some of these cases. Okay, that ends what I wanted to kind of explain to you in this podcast. Good luck, and let me know if you have any questions. See you tomorrow. Bye.